Start recording, and we are now recording. Thank you very much, James, and it's all yours.
Great. Well, Kate, thanks a lot for having me here. It's really a pleasure. I mean, I've been paying attention to the Build a Cell community for a while now, and it's really nice to have a chance to come here and talk with you guys about what we've been up to. Okay, so I guess I'll give you a little background of who I am. I have had a bit of a weird background to get to where I am now. I originally studied geology as a bachelor's student, and I was very interested in the history of life and on Earth. And then in graduate school, I wanted to get a bit more into chemistry. So I went to a graduate program between MIT and Woods Hole called the Joint Program in Oceanography. And I studied chemical oceanography. But actually, I was working with my two advisors were organic geochemists. So they were Tim Eglinton and Roger Summons. And that's where I sort of got my first interest in lipids. This was all work aimed at trying to understand how lipids could tell you about life, ancient life and ancient environments. And then I was working on this one kind of lipid called hopanoids, which are sort of like sterile analogs, and they're found in really ancient sediments. And that's what drew me to Dresden, because I found 𝗘𝗍𝗃, who's well known for his work on sterols and lipids and their role in membrane organization. And so I had a chance to come out to his lab to try to study what hopanoids do using the tools they developed in the Simmons lab. And then that was here at Dresden. And then this job at the university opened up at the B-Cube Center for Molecular Bioengineering 4 years ago when I started my lab. And so I was lucky enough to have a chance to start up a lab. And in this, I really decided to go completely all into the biological end of things and start trying to understand, what lipids do in cells. So that's sort of the brief intro to my biography. Let me share my screen here, and I'll get the presentation started. And let's see, let's try this. Okay, so now we got that going, and we go up here. It takes a second. Okay, now. All right, is the presentation up on the screen?
Yes, it's perfect.
Great. So the title is Exploring the Design Principles of Living Membranes. But we're actually, we're not trying to encompass that entire topic. We're actually working on a part of that problem, which is lipids. We're interested in the role that lipids play in membranes. And one of the longstanding questions in the field has been, why do cells need such complex lipidomes? Why do we need so many lipids? And another question that we're interested in trying to understand is, well, how many lipids does a membrane need to be a biomembrane? Okay, so I'm going to start off with a bit longer introduction than I normally give, because lipids aren't a topic that everyone's familiar with, and I want to make sure we all get on the same page. And then I'm going to discuss three stories. One's published and two unpublished in progress stories, and then a quick wrap up at the end. So I'm aiming for about 45 minutes in case you start getting antsy. Okay, so before we get to the lipids, let's get the boring stuff out of the way. What is life and how does it work? That's, I guess, sort of one of the major motivating questions here. Okay, well, I'm showing a picture here. This is from the Martin Scorsese movie Hugo, about a boy who finds an automaton, and there's an adventure about that. The automatons are a really interesting example of people trying to understand what is life. Hundreds of years ago, watchmakers and people that were making fine instruments were toying around with trying to build really intricate machines that had lifelike properties, motions and behaviors. And we've come a long way since then, and now there are very advanced machines that are even responsive and have facial expressions and things. Okay, but now we're trying to go a step further and replicate this process with biomolecules. And the whole thing comes down to, is life something we can really think about building or engineering if we have a complex or clever enough system, or is it something unimaginably out of our reach? Okay, so if we think about the challenge now making biomolecular automatons or cells, we've come a long way in the last 100 years. So the basis for the genetic code, the structure of DNA has been worked out. How that code is translated into parts is fairly well understood, and how many of those parts interact in cells in metabolism and signaling networks there's a good body of knowledge on that. So we have all of these components and understanding of how they're interacting. But still, we haven't managed to, from the bottom up, put all these components in a tube and have a robust self-replicating cell come out. So we're missing something, clearly. What are we missing that's preventing us from reaching that goal? We're missing something that would allow us to explain, for example, how life could have emerged from a prebiotic world, how cells coordinate signaling in space and time, and how multicellular organisms can orchestrate development. And one of the key things that we're missing is an understanding of cellular organization. How do cells make sure that all their parts are in the right place at the right time? Now, there are many facets to cellular organization, and membranes are one of the important ones. So the take-home message from this part of the introduction is, if we eventually want to be able to build a cell, at some point we're going to have to figure out how to build a membrane. Okay, so let's think about how membranes are involved with life. What's their role? Okay, we go back to this question of what is life, and one way we can define life, there are many ways, is as a homeostatic microenvironment. So it's a, you have a cell and it's trying to maintain its physical and chemical properties within the cell using homeostatic mechanisms. Okay, and then you have an external environment, and the cell on the one hand has to protect itself from that environment, from pathogens and chemical stresses. On the other hand, it has to retain the ability to interact with that environment in order to signal with its neighbors or to take up nutrients. Okay, so there you have now this differentiation between the life, the inside of the cell, and no life, the outside of the cell. And mediating that interface is the membrane, which we can think of as a responsive interface. And I think responsive here is a key word. It's not just a brick wall, it's a dynamic material that can allow a lot of things to happen. All right, so one question is trying to understand what's the basis for this responsiveness. Okay, now we have some idea of how the membrane fits into the cell in general in a very simplified way. And now let's look a bit closer at the membrane itself and try to think about what are the problems a cell has to manage in order to build its membrane. Okay, so if we look on the left here is a beautiful animation by 𝗺𝗺𝗺𝗺𝗺 showing a depiction of the typical fluid mosaic model of the membrane. You have lipids forming a lipid bilayer, and in that bilayer are inserted proteins, integral membrane proteins. Now, the proteins carry out a lot of the functions, the transport and signaling. But in order for them to function properly, the material properties, the physical properties of that lipid bilayer have to be well regulated. They have to be kept within a certain range so that those enzymes can behave properly. So things like viscosity, lateral tension profiles on the membrane are all important, and many more properties are important for allowing proteins to all work properly together in this one two-dimensional film. And the way that the cell can dictate the material properties of the membrane is by deciding which lipids go into the membrane. So the lipids play a really important role in this challenge of making sure that the proteins function properly. Okay, now if we look on the right, there's a video that a colleague of mine, 𝗍𝗺𝗺𝗺𝗺, made, and it's a synthetic vesicle, and it's about a lipid vesicle, and it's about to be pulled from the bottom here. If you look at this bit here, he's pulling it with some molecular tweezers. And you can see how incredibly far you can stretch a membrane without compromising its integrity. And the point of this movie is just is an example to illustrate how mechanically stable a membrane can be. And that is one of the things that a cell has to also pay attention to. It has to make sure that the membrane has the right properties so that it's mechanically stable enough to withstand all of the different conditions that the cell has to face in its environment. So taken together, these two panels illustrate the two sort of most basic challenges that a cell membrane, that a cell has to meet in order to build its cell membrane. And on the one hand, it has to modulate the physical properties of the bilayer to maintain bioactivity. And on the other hand, it has to do that within the constraint that those properties allow the membrane to still be mechanically stable. And lipids are the key determinant in defining the membrane's properties. Okay, so now let's think about this in a schematic here. So I've depicted on the left, there's membrane fluidity, and on the right, membrane rigidity. And in some ways, these are kind of competing concepts or properties in the membrane, in a very simplified way. Okay, so what we're depicting here is that life wants to have its membrane sort of in an optimal range for rigidity and fluidity are optimized so that you have bioactivity but stable membranes. And the reason is, of course, if your membrane becomes too fluid, we go up to the top here, it becomes mechanically unstable and its barrier property fails and your cell falls apart. If the membrane becomes, the fluidity becomes too low, it eventually becomes a solid or a gel membrane. And that's where life stops because you no longer have diffusivity. So life is trying to, living membranes are trying to maintain their properties in some optimal window. Now, on top of all that, not only do they have to maintain their properties in a narrow window, but they have to do that while everything is changing around them. Temperature and pH and physical forces and et cetera. So, okay, let's imagine that problem. So we have a functional membrane depicted up here, and there's a very crude channel that's drawn here. Now let's imagine the temperature decreases. Now what happens is that the lipids become more tightly packed. The tension that's experienced by this channel will increase the force that's exerted on it. And we can imagine that a channel might suddenly not be able to open anymore because the forces are too high that are exerted on it. So that's not good. The cell, however, can sense that something is wrong with the properties of its membrane, and it can respond to that by introducing new lipids into the membrane to restore optimal properties, even though the temperature is reduced. So this is a process called homeoviscous adaptation. It's been documented for decades. However, despite being well described phenomenologically, relatively little is known about how this works, which lipids are required for homeoviscous adaptation, what are the patterns of adaptation, and what are the regulatory mechanisms? And finally, how do cells even sense these changes to begin with in the membrane? Okay, so let's take a second now and think about how lipid structure, so okay, for a starting point, lipid structure determines the properties of the membrane. Let's think about how that's relevant for life. Okay, so here we have a very simplified scheme showing fluidity versus stability. And we can imagine starting here on the right with a very simple lipid. It has very short saturated acyl chains. There are no double bonds. And this is a fluid, but this will form a fluid, but very unstable membrane. Now what we can do is we can increase the chain length. And as we do that, fluidity will decrease. However, stability will increase. So let's examine this space now with respect to what life wants. If we look at the lower part of this schematic, this cartoon, this isn't good for life because although it's fluid, we have unstable membranes, so it's not good. If we look at the left-hand side of this quadrant, this plot, we see that it's stable, but at some point when the acyl chains become too long, the membrane becomes solid, so no life. Right, so life wants to be somewhere over here in this region where you have stability and fluidity. How do we get there? Well, there are a number of different ways. One thing you can do is you can add double bonds to the acyl chains, and that allows you to slide over here and retain some of the stability while making it a fluid system. Another solution would be to add a different kind of lipid. So I've depicted here a very crude drawing of a sterol. Sterols can interact with saturated lipids to fluidize them, and then you can get an even more stable but fluid membrane. And this is a very important mechanism that eukaryotes and some bacteria use to create their surface membrane. Okay, so this looks very simple. I mean, we have on the page here 6 different lipid species, basically, or 5 different lipid species. Why doesn't life make it this simple? Why don't cells make it this simple? I'll tell you in a second about how complicated real lipidomes are. So now, knowing that lipidomes are actually far more complicated in the scheme here, and the fact that the diversity of lipid structures across the 3 domains of life is huge. Bacteria are as different from each other as they are from eukaryotes. So there's a huge diversity of lipids in each different organism. Is there any convergence in the evolution of lipidomes in different organisms? And can we explain that diversity with a common set of unifying principles? Okay, so those are some of the motivating questions as we get farther down the line here. All right, now I mentioned that lipids are very complex, and we're trying to get it to understand something about the principles underlying how lipidomes are assembled. Let's look at how the complexity plays out over different cell types. So I've shown here a scheme. There's a vertical axis and log scale showing number of lipid species from 1 to 1,000. If we look at eukaryotic cells, they're pretty complicated. They have hundreds of lipid species per cell in a lipidome. So those aren't very good model systems to study sort of the simple fundamental principles of the lipidome. Now, if we go to prokaryotes, bacteria, for example, can have much simpler lipidomes. So it was shown recently in a paper last year that E. coli has just over 100 unique lipid species. We call different lipid structures species. Okay, so that's getting better, but it's still not quite as simple as we would like to be, to have an experimental system where we can, through a reductionist approach, try to understand these principles. And going a step further, eventually we want to get to a level where we could have a very simple lipidome that could potentially be engineered from first principles to contribute to a synthetic cell. And also we might want to think about how complex the lipidomes might have been that would have led to the emergence of life, to cells with membranes, whenever that happened. Okay, so over the course of this talk, we're going to try to work our way down from E. coli, down from over 100 lipids down to 1 lipid, and see what we can learn along the way. So the first thing I'll do is I'm going to tell you about a recent story that we published. I'm not going to talk about the whole paper. I'm actually just going to show one conclusion from this paper that's important for this discussion. So we started thinking about going to bacteria and studying how complex their lipidomes are and how they deal with adapting to varying environmental conditions. So we started with this organism, Methylobacterium extorquens. It's a plant-associated bacterium. Now, why did we pick this organism? A little bit of serendipity. We had been studying it in order to understand the role of 𝗺𝗺𝗺𝗺𝗺𝗺. These are sterile analogs in bacteria. And this was a great system to do that in. So we spent a lot of time working with it and working out protocols to study this organism. And so we thought, well, let's take a look at this and see what its lipidome looks like and how it adapts. As it turns out, it's also a very good organism for this kind of study because since it lives on plant leaves outside, it has evolved to adapt to a broad range of conditions, diurnal temperature cycles of 20 degrees, chemical changes on the leaf surface, light, etc. So we did a study where we took these cells, grew them over in changed conditions like temperature, growth stage, salt concentration. We switched the carbon source from sugars to methanol, and we looked at the effect of adding detergents. Okay, so here's a graphical abstract of the paper, and just to give you a quick overview. So we started by growing cells over varying conditions, and that's work that Sander Risk and Daniel Grosser carried out. And then we took those cells and we analyzed their lipidome, and that's work that 𝗵𝗇 and Oksana Lavrinenko did. And then we analyzed the lipidome and thought of different ways to parse the data so that we could extract some of the patterns in lipidome adaptation. And that's work that Magda Ryczynska and also 𝗵𝗇 contributed to. Then we took some of those patterns and we asked, what is the biophysical implications of the way that these lipids are being remodeled? And we learned a few new things. I'm not going to talk about that today. And then finally, Helena Jambor, so that was work that 𝗃𝗍𝗃, a postdoc in the lab, did. And finally, Helena Jambor was a data visualization expert that came in and helped us tremendously coming up with the final figures and visualizations for this study. Okay, so first thing we did was just ask which conditions led to the most change in the lipidome. So one easy way to do that is we can do a principal component analysis of the lipidomic data. So principal component analysis, or PCA, is a tool that you can use to simplify a multidimensional data set. So you can express the variance in that data set in 2 dimensions, even though the data set itself has many dimensions. So the input in this case is the lipidome, the abundance of all the different lipid species for cells grown at all these different conditions. And the way to interpret a plot like this is that the distance between 2 points, 2 samples in this case, reflect the difference in the lipidomic composition. So we can basically use this as a quick readout to see which conditions had the most effect on the lipidome. And what we can see is, here's the standard growth condition in red. And clustered around it are most of the experimental conditions that we tried. So this indicates that most of the conditions didn't generate a very large change in the lipidome. However, three conditions did give a relatively large change. So high salt concentration, when the cells went into stationary phase, so when they stopped growing, and temperature had the largest effect. So that right away gives us sort of a map to then zoom into certain conditions and ask, okay, now how is the lipidome actually changing? So, and one of the questions is actually how many lipids are required to adapt, for the membrane to adapt to these different conditions. So to ask that question, we have to now actually look at some data. So this is a table. with some data in it, and it's basically showing you all of the lipids we measured. So we have on the right-hand side, these are the lipid abundances. So each dot reflects, each row is a lipid species, and each dot reflects a sample, a replicate of a sample for each condition. And the red dot is the cells grown at the standard condition. And we have here the whole phospholipidome and 2 hopanoids that we measured, but I'm not going to talk about those today. So we cover over 90% of the lipidome of the cell. What we're missing are the lipopolysaccharides, which are an important lipid in the outer membrane. Okay, so let me break this down now. I've grouped this data according to the structural features of the lipids that are important for dictating the properties of the membrane. So there's 3 important structural features here. The first are the kind of head group that the lipid has. So if we look over here on the right, I've depicted a very crude drawing of a lipid. And what you see is the thing that they all have in common in terms of the head group is this glycerol with a phosphate group attached to it. And attached to that are 2 acyl chains. These are the hydrophobic parts of the lipid. Okay, so they all have that in common. Now, what varies is the chemical structure attached to that phosphoglycerol group. So you can have another glycerol, that's phosphatidoglycerol. You can add an ethanolamine, choline, or you can have a lipid with 2 glycerols, cardiolipid. So these are the 4 different head groups or classes of lipids that we detected in this organism. Next we have the length of the acyl chain. So here we depict it as the cumulative length of both of the acyl chains. And in this organism, it makes acyl chains with 16 carbons and 18 carbons. So that means we can have 3 different types of chain lengths, 32, 34, and 36. And those are shown over here. Next, we have the number of double bonds per lipid, or the level of unsaturation. And in this organism, we showed that there's 2 positions that can be unsaturated. There's a position here at the delta 11, 11 carbons from the carboxyl group here. And then there's also one at delta 5. So that means that you can have up to 4 double bonds per lipid. All right, so we discuss these as structural features of the lipidome, and they're very important because these are the things that are really affecting the properties of the membrane. Now, okay, we asked the question at the beginning of this, well, first of all, just make note of the fact that the number of lipids we've detected here, covering more than 90% of the lipidome, is about 30, 27, 25 phospholipids, 2 hopanoids. So that's already one of the simplest lipidomes that's been reported. Now, how do we evaluate which lipids are changing and how many of them? Okay, one thing we can do is just assess the variance of each lipid species, either over all the conditions or over a subset of conditions. And we can do that by taking the difference in the abundance from the max and min, or we can take the standard deviation or variance. And in any case, all those approaches give qualitatively similar results. So we can do that, and then we can see that some lipids vary not so much and others vary a lot. And now we can ask questions using this information. We can either sum that up and use that as a measure of total lipidomic variability, which we'll come back to later in the next project. Or we can actually now evaluate how many lipids are variable and how many aren't. And that's what that's the main point I want to get out of this particular study that we're discussing. So how do we do that? Let's take the lipidome. This is the same data as you saw on the table, but now we've reordered the lipid species. Now this is shorthand over here. This is the head group, chain length, double bonds. So this is shorthand for the lipid species structure. We've reordered all the lipid species according to their mean abundance. So from high abundance to low abundance. And the gray bar here is showing the range of abundances for each lipid species over all of the conditions. Okay, so first we ask how much is each lipid species varying? And in this case, for example, we take this lipid 𝗚𝗃36.2. So just as an example, we look at, for example, the standard deviation of the abundances of that lipid over temperature and growth stage. We get a standard deviation, and then we can use that as a rough measure of variability. And in this case, we plot that as a heat map going from yellow to red, low to high. Okay, so now we make a heat map of all of the lipid species for each of the conditions that we tested: temperature, growth stage, salt, detergent, and methanol. Okay, so one of the first things I think you can appreciate from this heat map is that most of the lipids aren't varying much at all. And the ones that are varying are mostly the high abundance lipids, the ones that are more abundant. However, there's some exceptions. So here we see with temperature change that these lipids with lots of double bonds are quite variable. And that makes sense since double bonds are one of the main tools that cells use to adapt to temperature. Okay, now we can ask how many of these lipids account for most of the variability? during adaptation. So to do that, we asked how many lipids account for 90% of the total lipidomic variability for each of these conditions. And when we did that calculation, we came to numbers ranging between 11 and 13 lipid species. So that means that out of the 27 lipids we measure here, less than half of them are involved in adaptation. So this is an interesting number, especially if we're thinking about building membranes. So in this case, this provides an upper limit on the maximum number of variable lipids that you would need to assemble an adaptive membrane that could adapt to all these different sorts of conditions. So we now know that 11 lipids maybe is an upper limit on the number of variable lipid species you would need for an adaptive synthetic membrane. Okay, so that's really all I want to say about this study. There are a couple interesting points, probably the most interesting points of the paper I didn't discuss, but if you really want to hear more about it, I just gave a talk last week, a 30-minute talk specifically on this paper. And if you click on this, the barcode there, it'll take you to the YouTube link and you can hear more. But the key points here is we examined the lipidome of Methylobacterium extorquens. We showed it has now one of the simplest lipidomes that's been reported so far, 25 phospholipid species. And as few as 11 lipid species are involved in adaptation of this organism to varying conditions. All right, so let's go back to our scheme here. Now we've gone from E. coli, just over 100 lipids, down to Methylobacterium, which is around 30 lipids. But there's still a lot of lipids, a lot of things to think about. Can we start to get even farther down the scale here and approach 1? Well, when I started the lab 4 years ago, I started thinking about that and what would be a good system to try to address that challenge. And one of the things that came up, going back to the old literature from the 60s and 70s, was mycoplasma. And there was a lot of work done by 𝗍𝗪𝗺𝗀𝗻, who was one of the godfathers of mycoplasma and membranes. Okay, what are mycoplasma? These are small pathogenic bacteria that live in mammalian systems. Well, they're also insect and plant mycoplasma, but the common ones we think of are mammalian pathogens. They're some of the simplest bacteria that you can cultivate in the lab. So some of them, the synthetic cell, the minimal cell, which is a synthetic derivative of mycoplasma, has fewer than 500 genes. They're very simple in terms of their structure. So they have one membrane, no internal membranes, and there's no cell wall. So basically, it's like a liposome, a synthetic membrane with a cytoplasm. So for us, it's experimentally a really terrific system. And one really important aspect of the mycoplasma that was especially attractive for us in terms of studying membranes is the fact that they don't really synthesize their own lipids. So they get all of their lipids from either the host that they're infecting or the media that you grow them in. So that means that we can experimentally control the lipidome of the mycoplasma just by changing what's in the media. So I thought that sounds like a really exciting possibility. And that's when I got in touch with the folks over at the Venter Institute, the JCVI, John Glass and Kim Weiss and many, Clyde Hutchisons and Ham Smith. And they were really helpful, and we've been having lots of discussions with them, and they've really been a big help to us getting into this world of mycoplasma and the minimal cell. Okay, so we started off on this adventure. This is, by the way, an EM image we took a while ago of the minimal cell dividing, and I like it because it has these little ears sort of poking out of the edge there. Okay, so the work I'm going to talk about is all unpublished, and it represents some of the key findings we've made in the past few years. And it's all been done by 2 PhD students, one Natalia Safronova, who's now approaching her 4th year and beginning to write up her thesis, and then Isaac Justice, who just began a month ago. And so they're spearheading this initiative. Okay, so let's start with a question: where do mycoplasma get their lipids? There's 2 ways. One is, okay, I lied to you a little bit. I said they don't synthesize their own lipids, but well, they don't really, but they kind of do. So let me explain what I mean. If they can take fatty acids up from the external environment, from the medium. So for example, they really like to have, they need, in fact, C18-1, so a fatty acid with 18 carbons and 1 double bond, and also C16-0, so that's oleate and palmitate. So if they take those fatty acids up, and they can synthesize the glycerol head group, and using the fatty acids in the glycerol pathway, they can synthesize phosphatidoglycerol and cardiolipid. Okay, so that's all they can do in terms of lipid synthesis. The other way they can get lipids is they can take up lipids from the media or the host, so preformed lipids. And what they need absolutely is sterol, so cholesterol, typically. So they absolutely have to have cholesterol to grow, and they take that up from the environment. But it's also been shown that you can feed them phospholipids or sphingolipids, and probably many other possibilities. So they will take up lipids from the environment, put them in their membrane. Okay, so now you have an idea of where they get their lipids from. First question we asked was, well, okay, what does their lipidome look like if we just grow them on an undefined lipid diet? So historically, the way people do that is they grow these cells and they use animal serum as an undefined mammalian lipid source. So we did that. We took the cells. In this case, I'm showing you data from M. mycoides, Mycoplasma mycoides, and JCVI Syn3.0b. And this is the synthetic minimal cell derivative of M. mycoides. And what we did was we did a lipidomic analysis on those cells grown on this undefined mammalian lipid source to ask which lipid classes do they actually take up and in what abundance are they? Okay, so you can see here there's the abundance in terms of mole percent, and these are the lipid classes: so cardiolipin, 𝗵𝗃𝗃choline, 𝗵𝗃𝗃glycerol, sphingomyelin, and cholesterol. So now we have for the first time a quantitative look at how the classes of lipids are distributed in these two minimal simple organisms. And so that's already a really nice starting point. And now we've been having some discussions over the past years with 𝗺𝗶𝗃𝗃𝗃𝗃's group. They recently published this paper in eLife modeling the whole cell of the minimal cell. And we're now discussing how we can contribute these lipidomic data to improve the models in terms of the lipid balance. And we're now thinking about how this can go on to the next model. So, and we've had a lot of fun discussing over the past year. Okay, so now that we have a rough picture of which lipid classes these organisms take up, then one question to start with is the lipid uptake selective, or are they just taking up whatever happens to be there? So one way to address this is to, again, let's use a PCA analysis. We did lipidomics on the media, and we did lipidomics on JCVI Syn3.0 and mycoides. And right away, we can say that based on a PCA analysis, the lipidomes, the lipid composition of the cells is different from that of the media. So this strongly indicates that they are selectively taking up the lipids. Another question is, do mycoplasma remodel their lipidome with temperature? So are they actually able to selectively adjust which lipids go into the membrane as conditions like temperature change? Or do they lack that ability since they have evolved to grow in mammalian systems with a controlled temperature? And again, we can use PCA to answer this question very broadly. And what we see is each of these dots are the cells growing at different temperatures. And we see clearly that they do, in fact, adjust their lipidome with temperature. Okay, so now Natasha started her PhD project with the goal of now saying, okay, can we now define the lipid diets in a way that will allow us to ask questions about how the complexity of the lipidome influences the function of the membrane and ultimately how the cell can adapt to different environments, its viability. So the logic behind Natasha's project started with just trying to define different, establish different defined lipid diets that we could grow the cells on. So we know that they need cholesterol, so that has to be in every diet. So that's a given. Now, the simplest lipid diet that you can grow them on, which was shown a long time ago, is you can give them cholesterol and 2 fatty acids, palmitate and oleate. And this represents the kind of minimal defined diet that you can grow mycoplasma on. Now, what Natasha wanted to do was extend that and begin to ask, okay, so if we now add in, let's say, a phospholipid, what does that do? Now, let's instead add sphingolipid, sphingomyelin, and see what that does. And then let's add all of them together and see what happens. So we're systematically increasing the complexity of the lipid diet. Okay, and so the first thing we did was just ascertain that, in fact, by altering the diet like this, we can control what lipid classes end up in the cells. So here we're looking at lipidomic data showing the classes in the cells that were grown on the simplest diet, which is cholesterol and 2 fatty acids. And you can see that there's only cholesterol and PG and cardiolipin, and both of these they can synthesize from fatty acids. Next, we look at the more complex defined diet. So first we tried PC plus 2 fatty acids and cholesterol, and sure enough, we see that they take up PC, quite a lot of it. Next, we ask, okay, let's feed them sphingomyelin. And sure enough, we see that they have sphingomyelin. Now we add both, and we get a cell that has both sphingomyelin and PC. So proof of principle works. We can now dictate which lipid classes are in the lipidome and begin to explore what effect that has on the membrane of the cell. Okay, so the first thing that we asked is, how does the complexity of the lipid diet affect the ability for the membrane to adapt to temperature? So going back to our concept of homeoviscous adaptation, let me just briefly explain you what we would expect to see. Let's imagine you have a cell and it's grown at 37 degrees and we measure its membrane viscosity and it's here, and that's its optimal viscosity. Now we change the temperature to 30 degrees. If there's no adaptation, we would expect the viscosity to increase, and that would be that. However, if the membranes are adapting to temperature, we would actually expect them to try to restore the viscosity to something close to what they had at 37 degrees. Now we can plot that a different way, which might be easier to read out, by saying, okay, if we take the difference in viscosity between the two temperatures, in this case, we're looking at 37 and 30 degrees Celsius. If the cells don't adapt, then we see, in this case, a negative value. So this is no adaptation. If the cells do adapt, the value should be close to 0. All right, so let's see what happens when we do this with real cells. So right now we're looking at a plot. On the vertical axis, this is, we measured viscosity using a probe called DPH, and we measured its fluorescence anisotropy. And this is a parameter that's very closely coupled with viscosity in the membrane. So as a control, we just took synthetic membranes, liposomes, and we measured the viscosity difference between 37 and 30 degrees. So this is more or less what we'd expect for membranes that aren't adapting. Okay, and this red line sort of shows where we'd expect to see things if they are adapting. Now, first we take the cells with the simplest diet, 2 fatty acids, and they're terrible. They even seem to do worse than the dead membranes, and we'll maybe come back to why that is later. Okay, so next we add in, Natasha adds in PC, so a phospholipid. Well, they're still not doing very well. It's pretty much the same as the liposome control. All right, now we add in sphingomyelin, and again, we're not seeing that they're adapting very well. Okay, so now we add both, and that's where we got a surprise. If we add both PC and sphingomyelin, we see that the membrane can actually adapt to temperature. That was cool. Then we tried one more thing. We added some more fatty acids. So no phospholipids, just fatty acids. We added 4 different fatty acids. And there we also showed that the membranes could adapt. So what's going on there? Let me explain to you what we did with the fatty acids. First of all, let me explain why 2 fatty acids might be a problem for the cells adapting. We give them palmitate and oleate. So you have a long chain with a double bond and a short chain with no double bond. Now let's imagine what happens when the cell is trying to adapt. So we have this scheme of viscosity, and we start with a lipid that has both of these fatty acids. Now let's imagine the temperature drops in this, sorry, forget temperature. We're sticking with a constant temperature, but we're changing the lipid composition by modifying the acyl chains. So let's imagine what happens when now we remove the saturated chain and add a chain with a double bond. Well, in this case, we add a double bond, so that should reduce the viscosity. On the other hand, the chain gets longer, so that should increase the viscosity. So these two things cancel each other out. Probably not exactly, but they're competing with each other. Okay, let's imagine now instead we replace the chain with a double bond with a chain with no double bond. In that case, what we're doing is we're removing the double bond, but we're making the chain shorter. So again, we have 2 things that are going in opposite directions. So the cell is fighting with itself to try to adjust viscosity using these 2 fatty acids. And this may explain why, in fact, while it's adjusting, it seems to actually go in the wrong direction. Okay, so we thought, well, let's try something different. Let's add 2 more fatty acids. So the problem before was that double bonds and chain length, 2 structural features which can affect the viscosity of the membrane, were coupled. You can't change one without the other. So we thought, let's try to decouple those by adding a long saturated fatty acid and a short unsaturated fatty acid. Now what the cell potentially can do is it can, it can replace the short saturated chain with a short double bond, short unsaturated chain, or it can replace the short saturated chain with a long saturated chain. In this case, it can either increase or decrease viscosity. So just by adding 2 additional structural features, we're able to go from disaster, no adaptation, to perfect adaptation. So this really demonstrates how the structural degrees of freedom of the lipidome directly impact the membrane's ability to function, in this case, to adapt to temperature change. And so we have a very clear-cut case showing the relationship between lipid structure and membrane function. Now, a prediction that we had from that now is that if the lipidome doesn't have the right parts that it needs to adapt, it might be trying to overcompensate. It might have to actually adapt more in order to try to do something. Whereas if it has the right parts, the right fatty acids or the right phospholipids, in principle, it may have to do less work in order to adapt. And so here is a plot showing we've calculated the total degree of lipidome remodeling. Again, think back to that first story I told you about. We've added up the variability of each lipid species for each of the for a change in for cells adapting to temperature change for each of these diets, and we've plotted total lipid variability in terms of mole % abundance. And what we see is, in fact, that the two diets, the more complex ones that showed homeoviscous adaptation, also show much less total lipidomic remodeling, which is consistent with our prediction. So that was super exciting. This is all kind of coming together. Now, the problem is these systems that Natasha's looking at, even with the simplest lipid diet with 2 fatty acids and cholesterol, the lipidome complexity is actually not that much different from methylobacterium. We still have about 30 lipid species, and that's because they can synthesize all of these phosphatidoglycerol cardiolipin species on top of the phospholipids that we feed them. So we started thinking, okay, can we actually get around that and really define the lipidome and make it even simpler than 30 and get down to our goal of maybe eventually 1? Okay, so that's where Isaac Justice comes in. So he's just started his thesis project, and his goal is to try to define the lipidome, constrain the lipidome, and make it as simple as possible so that we can really begin to study what happens to membrane function as we go to this critical threshold of simplicity. Okay, so a first attempt at doing this, we thought, well, okay, let's take the minimal defined diet. And we know that the problem here are the fatty acids, because those go in and the cell can make lots of things with those. So let's, instead of giving them fatty acids, let's just try to feed them a phospholipid instead. Well, they grow on a cholesterol plus 1 species of 𝗺𝗺𝗺𝗀𝗃choline. So Isaac set up this experiment. Here we're looking at a thin layer chromatograph. So this is it's like a gel for lipids. Essentially, we put our sample here and we separate it, and each of these bands is a different lipid class. So this is a standard in the left lane, and the 2 important things to pay attention to is the cholesterol and 𝗺𝗺𝗺𝗀𝗃choline. And we're going to look at how changing the diet affects the distribution of lipids. Here are cells grown on serum, so an undefined lipid source. So they have cholesterol, they have sphingomyelin, they have PC, they have cardiolipin and PG, all this stuff. Okay, so Isaac took those cells. He now adapted them to growing on only one species of PC and cholesterol. And here's what he got. It looked pretty promising. You have PC, you have cholesterol. There's still a bit of carryover sphingomyelin from the serum. So he grew them a bit longer to adapt them more to get rid of this carryover. So this was after a few passages. Okay, so now we go over 5 passages and suddenly all these other lipids appear. What the heck is going on there? So there's PG and cardiolipin again, and this thing. Okay, so we thought, what's going on? Okay, well, this band down here actually gave us a clue. This is very likely lyso-PC. This is when you cleave one acyl chain off of the PC. And what we think is happening is that at some point they adapt and they turn on the tools that they need to now scavenge the acyl chains from the phospholipid that we feed it. So basically they're taking lipases and acyl transferases and ripping off the acyl chains and then using those to make whatever lipids they want. So that was pretty frustrating because we were so close to having a defined lipidome, and then mycoplasma outsmarted us. So then we started thinking, okay, can we trick the mycoplasma? Can we do something that'll prevent it from doing this? So the way that they're scavenging these acyl chains is that they're taking advantage of this ester linkage here. And they have enzymes that allow them to break those linkages to cleave off the acyl chains. So we thought, let's get a really similar lipid, same head group, same acyl chains and almost, except there's no ester group. Instead, we have an ether group, and these can't be attacked by the enzymes. So the cells shouldn't be able to break them down. And maybe using this, we can trick the mycoplasma. Okay, So Isaac did that. He grew the cells, adapted them for 5 passages, and we did it. We now have the cells growing with 2 lipid species. Now they aren't very happy. They're very unhappy, but they're growing, and Isaac has them passaging continuously. So they really are alive. So now we can go back to our scheme here, and we can say, all right, we have now gotten all the way, almost all the way to the bottom to 2 lipids. And this is fantastic because now moving forward, we can begin to add complexity back in and ask what happens to the membrane as we add in different lipid species in a controlled way. Well, is there a critical threshold, or are there critical thresholds where certain levels of functionality are achieved? And how does this affect the viable range of the cell itself, which is the important consideration for building a cell and designing a membrane for it? Okay, so that's what I'm going to tell you about the Mycoplasma project. And the last thing I'm going to tell you about is going to switch gears a little bit, and we're going to get away from living organisms, and we're going to ask if, going back to the origins of life or protocells, is it possible that a membrane made from one lipid could actually do anything useful for a living system? Could a membrane from one lipid have, for example, provided some fitness advantage to an early biochemical system emerging on a primordial Earth, for example? Okay, so this introduces a project, and this is the last thing I'll discuss. We're getting to the end, don't worry. That's a PhD student, Thomas 𝗘𝗅𝗺𝗃, has been working on, and he's very close to now getting a paper together. I'm just gonna show one quick highlight from this study. So the project is trying to think about how RNA interacting with lipids, what implications that might have for RNA activity and function. And in particular, can this give us some new ideas for how to design novel synthetic systems and potentially insight into the origin of life? So, okay, what are we talking about here? What you're looking at is a synthetic membrane, and it's phase separated. So there's 2 liquid phases. One is a liquid disordered phase, and that's shown in gray here. And the other is a liquid ordered phase. And actually the thing that's labeling it is not a lipid probe, but it's an RNA aptamer that's fluorescently labeled. We took this sequence from a study by János et al. Back in 2006. These are RNA aptamers. They're very specific sequences that show a high affinity for a particular target. In this case, they generated aptamers for phospholipid membranes. And in particular, they found some aptamers that seemed to selectively localize to more ordered liquid domains. So this is really cool. When I discovered this work, I thought, wow, this is really amazing. And so we synthesized these RNAs, and this is an image, this is an experiment we did just to convince ourselves that this works. And it really, in fact, this RNA selectively binds to the ordered phase and not to the disordered phase. So that's exciting because it shows that RNA has the capacity to potentially distinguish membranes with different composition and property. But it also means that RNA can bind to membranes, and that could potentially have implications for RNA activity and using membranes to modulate RNA activity. Okay, so Tomek wanted to explore this, but the problem is with the aptamers and liquid membranes is that the binding is very sequence specific. So you need a very specific sequence and presumably secondary structure in order to get this ability to bind to membranes. But one of the things that came out of all of these studies that were done by János and others in the past is that if you have a gel membrane, actually a lot of RNA will stick to that. In fact, all RNA will stick to that, at least all the RNA that was tried. You can take a random mixture of RNA, 40 more oligos or something, and throw it in the tube with a gel membrane and it all sticks. So we thought that's pretty cool. Let's explore this system and see if we can use that as a platform to study how binding to a membrane could affect the activity of RNA, and in particular ribozymes. These are RNA enzymes that can catalyze different reactions. Okay, now you might be wondering, well, who cares about gel membranes? Living cells don't have gel membranes. And you're right. Well, there are a few studies here and there that suggest that they might play a role, but that's all highly disputed, and we're not going to discuss that today. But okay, we're not talking about life, at least cells at this point now. We're talking about either RNA-based systems or potentially going back really far in RNA world, where you might have had a network of RNA molecules, and perhaps some sort of scaffold might have been useful for those. Now, in the context of the origin of life and the emergence of lipids and membranes, in the absence of the enzymatic machineries for making double bonds or sterols or hopanoids or whatever, in some ways you're kind of limited to things that have long saturated chains. And so you have this, we go back to our original scheme where you can increase the chain length. And as you do that, you go from an unstable system to a stable system. But it's a stable system that's not fluid. So in many ways, gel membranes are up here are the things that might have been most likely to accumulate. They're more mechanically stable. They're also more resistant to variations in chemical conditions. So these might have been the things that started off the role of membranes in life. I mean, we can argue about that. I'm not saying that's how it is, but it's one possibility. Okay, so that's why gel membranes might not be as irrelevant as some of you would have thought. Okay, so just a brief, I'm just going to talk about one result Atomic has out of many. It's just the tip of the iceberg here. So we started playing with the R3C ligase, which is developed in 𝗺𝗺𝗺𝗃𝗍's lab. And this is a two-component system. It's two pieces of RNA. One is a ligase and one is a substrate, and they basically react with each other till the two pieces stick together. And we can visualize that on a gel. Atomic made this beautiful gel. We can have the substrate here. That's this orange thing. It's a short fragment in the absence of the ligase, and there's no product. In this case, we're not showing the ligase. It's only the product and the substrate are fluorescently labeled. Then if you add the ligase, suddenly the product is generated. So it puts these two together and you get a longer fragment. So then so the idea was, okay, you have these things in solution and they're diffusing around. Now, if you add membranes and they bind to them, you can effectively up-concentrate them, and maybe that'll enhance the activity. So Atomic did this experiment, and sure enough, it works. So on the y-axis here, we're looking at the reaction rate of this R3C ligase. On the left is without membrane, and on the right is with membrane. And we get a 50%, more than 50% increase in the activity just by adding a membrane and making this thing bind to it. So this got us really excited, and Atomic is now in the final stages of putting together a manuscript, and hopefully we can put this out on bioRxiv soon, and you can get the full story in the near future. So pay attention to our Twitter account when we release it. But what this shows is that, in fact, a membrane with one lipid can, in principle, have value for a biochemical system. Maybe not a cell, but maybe. But for sure, if we go back to these simple primordial systems, we can imagine a new way in which a gel membrane with one lipid could have contributed to providing a fitness advantage to emerging RNA or other biochemical systems. Okay, so it's time for me to wrap up. Let's come back to the thing here. So design principles of membranes. And one of the things we're really focused on is their ability to adapt and be responsive. Now, look, I'm showing you some more data here, but don't worry, it's not going to go much farther with it. Here's a plot where we've basically measured the viscosity of mycoplasma membranes over a whole bunch of temperatures. And what we see is that they can adapt over a certain range. And then beyond that range, that breaks down and they can no longer adapt. So, okay, one of the big questions is really exploring, okay, what is that range? What is the range of membrane properties that can support life? And how does manipulating the complexity of the lipidome affect how well the membrane can achieve that range? And where does everything break down and cause things to stop working? Now, let's think about this. It's not a one-dimensional system. Viscosity is not the only important parameter. There are many properties of the membrane that are important for life. For example, permeability, phase properties, the ability to potentially phase separate for lipid rafts, for example, many other things. So we're now talking about a multidimensional space that the living membranes have to find a way to optimize all of these parameters. And most likely it's not a boring rectangle like this, but rather some more interesting shape like that. So what we're now trying to do is take these systems we develop and begin to explore what is this volume and what are the limitations in the relationship between the complexity of lipids and the ability for life to achieve this optimization process. And most importantly, for the problem of building a cell, how does all of this affect the range over which a cell can survive and adapt to different conditions? So, we started off with some questions at the beginning. Why do cells need such complex lipidomes, and is there any convergence? I don't think we've answered those questions, but we have shown how simple we can make a membrane and still have a living organism. And we're now in a position to really begin to address the bigger questions, which is why do we need so many lipids, and how few lipids do we need to engineer a synthetic membrane for a synthetic cell? Okay, so with that, I'd like to wrap up. And of course, I have to acknowledge my whole lab. It's really been wonderful working with them. In particular, also the university, the Technical University of Dresden, and the B-Cube Center for Molecular Bioengineering, which is my host institute, as well as the Volkswagen Foundation for funding one of our initiatives with Ilya Leventhal, Robert Ernst, and Maya Schuldiner, and the BMBF for very generous funding to get the lab started and keep it running, as well as many people who have provided really important feedback and discussions over the years. And we've been lucky to have input from all of these people and their generous thoughts. Also Lipotype deserves a big thanks for doing all of our lipidomic analysis. So with that, I'm going to wrap up and take any questions you have.
Thank you very much, James. That was an awesome talk. You have quite a few questions in the chat. If you would like to go through them, that might be a good.
Okay, let me find the chat here. Should I end the screen share? Do people usually?
I think unless you have a dual screen, you have to end screen share.
Okay, there we go. So now I'm back here, and we have some questions in the chat. So all right, let me make this a bit bigger so I can actually read this stuff. My vision's getting worse with age. Okay, so which is the first question here? Is the lipidome simplest because it's primitive, or it's de-evolved because of parasitic or other life conditions? Great question. I think a little bit of both. Well, it's not primitive. I mean, these are modern cells, and they've basically evolved from more complex cells to become less complex. So I think the second point of your question is more correct. They've lost a lot of functionality because they can rely on the host organism to provide a certain level of stability. Despite that, they still do seem to retain a lot of ability to adapt, and they're actually much more complex than I would have thought a simple minimal cell to be in terms of membrane adaptation. Okay, so let me go down the list here. I can look at my screen here. So another study that generated cells with greatly simplified membranes is B. subtilis, may be of interest. Membranes reduced to almost PG only. Yes, definitely. I mean, Bacillus subtilis is one of the great model organisms. And we toyed around with that for a while. And one problem we had with respect to the lipidomic analysis is that it's actually a technical challenge to do quantitative lipidomics using shotgun lipidomics, because that relies on distinguishing masses of lipids very precisely. And one of the main structural modifications that Bacillus uses to adapt to temperature, for example, is to change the isomerization of methyl branches in the acyl chain, so iso, anti-iso, and those can't be resolved by shotgun mass spectrometry yet. Just for the sake of getting started quickly, I sidestepped that, but yes, Bacillus is still a very important organism for understanding membranes, and that can't be understated. For building an artificial cell, if you're allowed to ignore constraints of modern biology, what lipids would you use to make the simplest, robust artificial cell? Well, that's precisely where we would like to end up, is to have a recipe, a cookbook. And you could say, all right, if we want a membrane for a cell that needs to do this, here's the 5 lipids you got to put in there. At the moment, I guess we're getting close to being able to give one answer to that question. Isaac has his thing with 2 lipids, and we're beginning to add lipids back into that to see when we go from, at the moment they're clumpy and they barely grow, when do we see them suddenly start behaving normally? I think that would give us a first answer. What is the lipids we would choose? Certainly something like cholesterol is important because it allows you to have a fluid membrane that's also very mechanically stable. Are there any hopanoids that can substitute for cholesterol mycoplasma? Yes, we've been meaning to try this, but actually it was done already back in the 80s by Kannenberg et al. And some others. They took hopanoids, they adapted the mycoplasma to them, and not all the hopanoid structures could support growth, but some of them could. So that's very interesting that it also supports some of my previous work showing that hopanoids are sterile surrogates. And that's something we will come back to in the future. In one of the earlier graphs, you showed the percent PC uptake appears to differ between Mycoides and Syn3. Why do you think that's the case? And is this strain of Mycoides from which Syn3 is derived? Yes, this is the same strain. This is all sent to us by the folks at JCVI. That's the strain the Syn3 is derived from. GM12. Well, why do they differ? Good question. I mean, we're starting to get into something I didn't talk about at all today, is trying to understand what are the actual mechanisms of lipid uptake and remodeling. So what are the transporters that are taking up cholesterol and phospholipids, and what are the enzymes that are remodeling acyl chains? So I can't tell you why yet, but hopefully in a few years, once we've maybe uncovered some of those mechanisms, we can begin to understand differences like the fact that minimal cell seems to really like sphingomyelin. Take-home message, life is a banana. Great talk, thank you. Oh, thank you very much. Let's see. Curious as to why the lipidome of JCVI Syn3.0 strain adapted the temperature in the opposite direction to the wild type Mycoplasma strain. Yeah, I mean, this is something that Natasha now is writing up in her paper, comparing looking into more detail into how the lipidome adapts. And there are some differences. The Mycoplasma, for example, seems to use more subtle changes in acyl chain remodeling, whereas Syn3 seems to rely more on class level adjustments. And so, that might eventually be explained by differences in their biochemical capabilities. So stay tuned. In the context of building synthetic membranes, as in designing membranes which can repair themselves, what is the role of membrane fluidity and lipid bilayers being able to close any discontinuities on the membrane? Yeah, I mean, that for sure. Actually, maybe a more relevant parameter would be intrinsic curvature. If there's a tear or something like that, the ability for the membrane to refuse. And things like, for example, lipids like PE might enhance that, reduce the energy barrier to having a membrane fuse like that. Also, having higher fluidity would, I guess, probably increase the rate at which a hole or something would be restored, just because you have more diffusivity. Interesting question. I hadn't thought about that much, actually. That's something to wake up at 3 a.m. thinking about. Final question. Can you quantitatively tune the composition of mycoplasma membrane with your defined medium? I.e., can the final composition be tuned by the concentrations you are supplying? Very cool question. That's something that Isaac now and Natasha are beginning to explore. And one way you can rephrase that is, how selective actually is the mycoplasma lipid uptake, and how robust are they to variations in lipid diet input? And we're now especially beginning to dig into that, since we know now that we can actually really control the input. And Isaac's starting to do some experiments to see, well, okay, if we vary the ratio of the lipids, how much does that actually dictate the ratio of lipids that end up in the lipidome? So I can't answer that question yet, but maybe the next time I give one of these, I can give you more details. Okay, so I think that's all the questions for today, unless anyone has any more they want to ask now.
Thank you very much. So I actually have a question, which is kind of related to me being a liposome person that actually does TXDL mostly. Have you looked at it from the point of view of which lipids for artificial cell could be the simplest to synthesize by biochemical reactions? Because that's what we really need. We need to make them. So
yeah. Well, that's one of the reasons why I was so excited about 𝗺𝗺𝗺𝗃𝗃𝗃𝗃𝗃, because they can in many ways do what sterols can do. At least they can interact with saturated lipids to form a liquid ordered membrane. And the 𝗺𝗺𝗺𝗃𝗃𝗃𝗃 pathway only requires one step from squalene. So you have to synthesize squalene, but you have one step and then you have a 𝗺𝗺𝗺𝗃, whereas cholesterol requires something 15 steps to get to cholesterol from squalene. So 𝗺𝗺𝗺𝗃𝗃𝗃𝗃𝗃 might be one solution for the sterol requirement. For the phospholipids, I mean, I think John Sutherland would be a good person to talk with about this. I mean, he's at the LMB in Cambridge working out these primordial chemical pathways for getting all the biomolecules, including lipids, and there's some cyanide-based reactions there that can explain how you might be able to get glycerolipids and other things like that. So maybe some of those things, eventually we should try to start putting in the minimal cell and see if they can actually support it.
Yeah, and I think you have one more question from Azim in chat.
Yeah. Can you elaborate on the phase differentiation of membranes using RNA? Yeah, I mean, what I talked about there was simply just liquid versus solid phase. So membranes that are in a liquid phase, in order to get an RNA to bind to them, you have to have a very specific sequence, an aptamer. When the membrane gets cooled off enough, or you change the lipid composition enough, you can get a solid membrane, a gel phase membrane, and that's where you start to get less selective RNA binding. Does that answer your question, hopefully?
Thank you very much, James. I really appreciate. I think that's one part of building a cell that we don't talk much about in the community. Like we say, okay, there's going to be a membrane, but this is crucial. We need to do it, and we need to find a way to build it. So I really appreciate your talk.
Yeah, thank you very much for having me. I'm a big fan of this whole effort, and it's fun to finally get to sort of meet all of you and have a chance to talk. So,
thank you.
Yeah.
So thank you very much. And if there's no more questions, have a great day, everyone.
Okay. Ciao.
Bye.
Bye-bye.